Bruker Fluorescence Microscopy Business Unit. I'm the Product Marketing Manager, and Jimmy is one of our software developers. And we're going to be chatting with you today about our Opterra Multipoint scanning confocal and some new functionality we've added with respect to spectral imaging in that instrument. Just to give you an outline of what we're going to cover today, we're going to go over a design overview of the Opterra, different scan modes in the system, and the light path, so you can understand the overall functionality of the system as a confocal imaging system. And then Jimmy's going to cover how we've been able to adapt that to allow some really interesting spectral imaging capabilities with the Opterra. Design overview on the Opterra. It's a super video rate high-speed slit scanner and can also be operated in a variable pinhole linear array field scanner mode. So it can operate as both a point scanner and a slit scanner. Uses an EMCCD camera as the detector. And we can get high-frequency scanning based largely upon the speed of the camera. Benefit of that is not only good temporal resolution as far as the data being collected, but we also minimize the phototoxicity of the samples. Some additional points here. The multiple scan modes allow for optimization of resolution, speed and light delivery. Can operate at a much higher frame rate in slit mode. If people want to gear their data collection to better resolution, they can choose one of the pinhole apertures available in the system. The other benefit of the in point scanning with the system is that the linear pinhole arrays and separate light paths on the excitation emission sides minimize emission crosstalk. Here's a picture of the aperture plate. This is a key design element of the system. You'll notice on the right there are several excitation apertures, and on the left several emission apertures. You'll notice that there's 3 linear pinhole arrays and then 4 slits on the excitation side, and then matching emission pinholes and emission slits on the emission path. The aperture positions, here's the different pinhole sizes: 60 microns, 45 microns, 30 microns, and then a variety of slit sizes. And this aperture plate you saw in the previous slide is controlled electronically by the software to allow the user just to pick for a given experiment which pinhole or slit size they'd like to use for the experiment. Quick overview of a schematic of the light path of the scanner. Input is coming in from a laser launch where we have visible lasers. Passes through a lens that shapes the beam into basically a rectangle of light that then passes through either a pinhole array or through one of the slits, reflects off of a mirror to a galvanometer that sweeps the image of the pinhole or slit across the field of view. The light reflects off of a polarizing beam combiner off a dichroic mirror out to the microscope, and then the emission returns to the scanner, showing in this green line, and you'll notice that it's now separated on a different level than the excitation path, so the 2 light paths have been separated at this point. Returns off the galvanometer, off of a mirror, back through the D-scan through the emission pinholes, reflects again off of a piezo, which then sends the emission out to the camera. Here's just a overview of the scanner itself, where you can see the light path laid out on the schematic of the instrument. Blue is the excitation path, and then green is the emission path returning out to the camera. So one of the major benefits of the system is because there is a variable selection of pinholes that the user can optimize the pinhole selection for the objective that they're using. And the goal is to choose a pinhole that allows the best out-of-focus rejection and basically matching up the pinhole size to the calculated area disk for a particular objective. For optimal resolution, as I mentioned before, you want to match the objective lens to be able to create a focal point that matches up with the pinhole size. And this allows a lot of flexibility for the user, where they can either optimize within experiments or having flexibility between experiments, so that choose a larger pinhole, get more signal, a little bit of loss of resolution, or choose a pinhole that's more closely matched up with the area disk. For higher speed types of applications, so the user can choose a slit. There's more signal, we get faster scanning. You do lose resolution in one dimension, but get a great increase in temporal resolution. And it's sort of a typical use case for users of this system. If they're doing experiments that involve high temporal frequency, they'll typically run in slit mode, but then when they want to be able to acquire higher resolution image data, they'll switch over to a pinhole mode. So another diagram that shows how the actual scanning works. So the red light is the light coming from the laser launch, shaped by Powell lens into a rectangular beam of light which passes through a pinhole or slit, scanned by the galvanometer. And if you look at this in the different type of schematic overview, during pinhole scanning, there's a piezo that's moving the aperture, the 32 pinholes in the pinhole aperture, in a vertical direction, while, you see there, while the galvos are moving in the lateral direction like this. So that during one frame time, the piezo will make one sweep, move the pinholes from one position to their starting position to being adjacent to the next pinhole, while the galvanometer is sweeping the aperture image rapidly several times during a frame time. And if you combine these two movements, you'll see this kind of a movement. So we're basically able to paint the entire field of view very rapidly, and then on the emission side, the emission return is also then painted on the camera chip to get an even illumination on the field, and then even return of the emission on the camera chip. So one of the benefits of the linear array that we're using is that we reduce crosstalk significantly compared to 2D array scanners like spinning disk type scanners. It's basically the square root with our linear array arrangement is the square root of the crosstalk of a 2D array scanner. This provides improved axial resolution, and see a diagram here to illustrate this. On the right is the linear array of pinholes that's in Opterra, and you'll notice that the crosstalk from any given, the crosstalk for any given pinhole is related simply to its two neighbors, whereas on a 2D array, the crosstalk of any given pinhole is a result of several pinholes around a particular pinhole. As far as detector and microscope choices on the system, user can pick detectors that are either oriented toward high speed. For instance, the cameras that are high-speed EMCCD cameras running up to 66 full frames per second with larger pixels, 16 micron pixels, or they can choose to use a higher resolution detector, slightly slower frame rate, but higher pixel resolution in terms of smaller pixels, something on the order of 6.3 to 8.88 microns in size. And the system can be run on either a inverted or upright microscope. Just a couple of data sets here to show the capabilities of the system. This is a imaging of zebrafish neurons, neurons labeled GFP, and this is showing a 100 micron Z range I'll play the movie again. You can see that there was signal throughout the entire 100 micron range. And another example of the imaging capabilities. This is a tumor slice labeled with CFP and GFP. Again, this is 100 micron Z range in a fairly dense sample. You can see that there's good signal throughout the entire specimen. So I'm going to now turn the presentation over to Jim, and he's going to talk about how we've been able to adapt the scanning capabilities of the system to be able to incorporate spectral imaging in the Opterra Multipoint scanning confocal. Jimmy.
Thanks, Jeff. Now that you have some background on our scanning system, we can move on to spectral imaging. So in the biosciences, specimens are more often than not labeled with multiple fluorescent dyes or genetically modified reporter proteins in order to study things like the interaction between anatomical structures or communication and signaling pathways or trafficking of different cells in response to stimuli. And so the traditional way that multiple fluorophores have been visualized is as such. One image is taken with a narrow bandpass filter that blocks everything except a tiny bit of the red emission, and then another image is taken that blocks with the bandpass filter that blocks everything except a tiny bit of the green emission, and the same thing with the blue emission, where another image is taken with a narrow blue bandpass filter. And the problem with this is that you're throwing away most of your signal every single time. It's also very difficult to do more than 3 colors. If you are doing more than 3 colors, the limitation of switching filters between each exposure is the main limitation of your temporal resolution. And finally, any technique to computationally unmix any overlap are really limited because you only have information for a small band of the spectrum. And so our goal is to improve upon this. And so the key to our approach is to use the compound prism that was first invented in the 1860s for astronomy, known as the Amici Prism. And the Amici Prism is a compound prism that's made up of several multiple prisms with the main design properties such that the center incident wavelength exits the prism parallel to the incident beam, which in this case our center wavelength was designed around a green wavelength. The other colors of light exit and refract around the center wavelength, depending on the properties of the different prisms and the differences between the interface of the prisms. So our design was optimized for linearity across the color spectrum of the bands so that the colors are spread out equally from the violet and red. And the main advantage of using the Amici prism compared to some grading technologies is that the transmission efficiency of the prism is close to 100% across the visible spectrum compared to around 40 to 50% for gratings. Gratings are often used in spectral systems because the dispersion that a grating can provide is often greater than a prism in order to span a greater distance between distinct detectors. But you'll see how in our architecture we don't really need that extra dispersion because our detectors are actually pixels on the camera which aren't spaced that far apart. For our design, we placed the prism in the emission beam path on the translating stage, as you can see here in the diagram, so that it can be easily put into a beam path for spectral imaging and can be easily taken out for normal confocal imaging. And so the way that the spectral imaging collection works is with the prism is as follows. The emission light from the 32 pinholes in pinhole mode that Jeff described enters the prism. The prism spans out the 32 points into 32 spectra. And the 32 points of spectra actually span entire row in the camera sensor. But as the galvanometer sweeps the 32 points across the sample, it creates a full spectrum from 32 lines that were scanned from the sample itself. for a single exposure, we now have the full spectrum from 32 lines of scanning on the sample. And in order to fill the 512 by 512 camera sensor chip, we need to repeat this 16 times between each exposure. We need to shift the scanning by one row so that we cover the entire 512 by 512 pixel spatial area from the sample. So to reinforce this a little bit further, we need to capture 16 images in order to make up a full spectral frame. So for each image, the piezo is stationary and emission is going through the prism, so we're collecting the full spectrum, but from only 32 lines on the sample, as illustrated by the red lines. So that's for the first exposure. For the next exposure, we move the lines down a row using the piezo and collect another full spectrum from 32 lines on the sample. After 16 exposures times the 32 lines, we've created a 512 by 512 image spatially with all the spectral components. So after collection, we're left with 16 raw images that we have to reconstruct into spatial data. And it's a significant amount of computation in order to process this raw spectral cube, but we've optimized the algorithm to do this in real time, meaning we're able to generate the spectral image for each wavelength as the data comes in. So we don't have to wait for all 16 images to be collected before starting the process. But instead, we're beginning with the first line of the first image, and we're able to piece together the spectral data into sensible spatial images. What this ultimately means is that we can live scan and have a live update of all 15 channels as the sample changes or the field of view moves, which gives the user interactivity and real-time adjustment of the data that comes in. And the final result is that we had an image of the field of view for each of the 15 spectral channels with intensity representing the amount of color in that color channel. So in some testing with the spectral mode, we wanted to verify the accuracy by simply testing a mix of color beads of known spectra. And so each pixel in the image now not only contains the total light intensity that was generated from that location, as you would get from a traditional grayscale confocal collection, but also contains information about the wavelength profile of that pixel. And so we're able to discriminate different colors of beads from just looking at the spectrum that each pixel generates. And so with this information, we can better unmix fluorophores that have overlapping spectra and are co-localized in the same area. And so there are projects underway in collaboration with university researchers to develop advanced unmixing and spectral processing software to better utilize a lot of this data. So there are techniques out there right now to be able to unmix fluorophores if you have known the known spectrum for a given sample, but we're also exploring ways to do some blind deconvolution where you would be able to unmix an unknown fluorophore or unknown spectrum from a fluorophore from a given sample. So we want to investigate whether needing to take multiple exposures for a spectral frame affected the quality of images, since it's possible that electronic noise from the camera could be additive after processing, and that what we found was that compared to a single exposure at the same time, there was no noise penalty in our signal and noise ratio when comparing the 16 or multiple exposures compared to the single long exposure time. We also really wanted to test the spectral resolution of the system. So we obtained some pretty challenging samples from Invitrogen, which are green-green beads that have one wavelength of green in the core of the bead and a slightly different wavelength of green than the shell of the bead. And under normal traditional confocal imaging with a green bandpass filter, we can't really tell the difference. They just show up as a uniform green bead. And as I mentioned, the core has a one green wavelength, which shows that as 525 nanometers, whereas the shell has a different green wavelength that's slightly less green at 512 nanometers. But in spectral mode, by using some preliminary unmixing techniques, we were able to reconstruct the images that showed this slight difference in green wavelength, with the core showing up, with just the core showing up in one spectral band, and then the shell showing up in another. And so researchers at the University of Wisconsin were especially excited by the capabilities and wanted to try their science with the instrument. And one researcher was really interested because in normal imaging of her Arabidopsis plant root, the spectra from chlorophyll basically overwhelms the fluorescent proteins that she's actually interested in. And so she's working on techniques to unmix the chlorophyll spectra from her spectral images in order to study trafficking and exocytotic events that have been hypothesized but have been never observed. We also have several researchers that are interested in zebrafish development, and the ability to multi-label their samples allows them to study developmental events as well as create these really beautiful images. So leveraging the high-speed spectral capabilities or high-speed spectral acquisition capabilities, we have researchers that can study cell division events in frog eggs and visualize mitosis with spindle formation and microtubules pulling apart the chromatid pairs in real time. And so let me play that movie once again, and you can see we actually captured one of the cells undergoing mitosis in this bottom left-hand corner here, whereas the other ones aren't currently in that phase right now, but we did capture one. So coupled with the stage movement of the microscope, we can create large field of view montages with full spectral resolution by stitching together multiple fields of view. So here's a montage that we pieced together of a zebrafish with the spine labeled with one color and then neurons labeled with another. So you can see, and also you can see the autofluorescence of the yolk sac here. That's coming back as a, I don't know how well it shows up in your screen, but it comes back as red, which wasn't labeled, but we can see the red autofluorescence, most likely from the blood. There are several researchers studying neural development and morphology during development, which these next two pictures show. And so this is a zebrafish brain with the neurons labeled in green. And here's another sample of zebrafish head, again with the neurons labeled in green. So we've integrated a spectral mode into our scanning and acquisition software, Prairie, that has features that allow you to interact with the data in real time. And so for each of the four image windows, the user can decide which parts of the 15-channel spectrum he or she wants to see and what color it maps to. And the choosing of the spectral channels here is interactive. So during a live scan, the user can turn on and off certain channels to visualize different parts of the mission. And so for instance, here for channel 2, we're visualizing something blue like a BFP label, and whereas in channel 4 we're visualizing a something more yellowish, something like a YFP. The way that the channels are combined can be switched between averaging and summing. So if you want to see more discrimination between the channels, you can sum the pixel intensities in each color bin versus average them. And this interface also allows you to toggle between spectral imaging and normal high-speed imaging with just a simple checkbox. So what really allows us to do this acquisition and processing for spectral mode is really the speed of the instrument. So we need 16 camera exposures for a single frame, so without the high-speed scanning capabilities of that TerraScan head, and also the state-of-the-art high-speed cameras like the Evolve Delta, acquisition would take way too long for meaningful biological experiments. We're also using a 512 by 512 sensor, but we're currently also exploring whether it's beneficial to use a higher resolution camera to acquire more spectral channels than the 15 that we're currently acquiring. But at its fastest rate now, we're acquiring about 4 full 15-channel spectral frames per second. And so let me play this movie again. So this is just a sample on the stage, and we're just moving around, and we're collecting and updating with full 15-channel spectral frame resolution in real time. So in summary, the advantages of our design are such that we can collect with negligible noise penalty, as we showed with the multiple exposures combining together. It doesn't really introduce much of any noise penalty. And also we can acquire with really high sensitivity. We're combined with the greater than 90% quantum efficiency for the cameras that we're using, plus the greater than 90% transmission for the prism that we're using. We're able to visualize signals that you can't get with any other spectral system. We get 15 channels of spectral resolution. We can capture and with really great temporal resolution. And with the design, we're currently able to switch between regular confocal and spectral imaging modes simply by dropping the prism into a beam path with a simple plunger, and just with a software switch, switching between confocal and spectral mode.
Okay, thanks, Jimmy. That's the end of the presentation, and we'd be happy to take any questions now. Okay. First question, how does the point spread function compare with point scanner or standard Yokogawa disk? Good question. With respect to point scanning systems, point spread function, assuming we're using a pinhole that matches a comparable size pinhole, is going to be fairly close to that of a standard point scanner. At this point, we've never had an opportunity to do a side-by-side comparison with a Yokogawa spinning disk. Next question. Is crosstalk worse in the axis of the scan than perpendicular to the scan? I'm not sure you have any idea what to go ahead, Jimmy.
So in our scanning, we're sweeping the galvos across the field of view, and also moving the piezos in the piezo axis at the same time, and the resolution should not be any different in either axis, I don't think. If that's the question, I'm not sure. So definitely in the z-axis there would be a resolution difference, but in xy axis it's...
Yeah, move on to the next question. Okay, next question. Has the instrument been used to distinguish fluorophores of similar emission, for example, GFP and FITC, or YFP and GFP? We haven't tried GFP and FITC. We've done some work recently with a group at the NIH where we're looking at 6 different fluorophores that were in the samples. These were all fluorescent proteins that were being expressed by the cell lines that they were working with. And in those cases, we actually were using samples that had CFP, YFP, GFP, M-cherry, apple, and Bodippy, and we were able to discriminate those 6 fluorophores in the samples. Question, has anyone attempted spectral fret with this system? That's not something we've tried yet. It's definitely something that we want to take a look at later this year.
Yeah, there are a lot of researchers at the university that are especially intrigued by the possibility of doing spectral fret, and they're really interested in trying it with our system.
Okay. What is the temporal resolution of the frog mitosis movie frames? Do you know what the frame rate on that was?
That wasn't, that was, let's see, they were taking, I think, 50 millisecond exposures every second or so. But if you wanted higher temporal resolution, you can definitely do it if your sample calls for it. We can get close to 4 full spectral frames a second if we needed to.
Okay, so the answer to that was that particular movie was that each one of the frames of the movie was at 1 second time intervals. It says it seems the scan rate is not sufficiently high, which causes a drift in the image. Actually, the drift in the image was in that mitosis experiment was actually due to the sample, not to the scanning itself. How do you calculate the spectral width on each pixel? The calibration of the in spectral mode is done by placing a reflective surface on the specimen stage, and we basically just then turn on the lasers that are being used. They're part of the system, and without an emission filter in place, we're able to just basically get back reflection off the reflective surface onto the camera, and we have a calibration routine that takes the emissions that we see on the camera from two laser lines and constructs a calibration curve from that. The spectral width on each pixel is basically, we don't really calculate that, do we?
Well, it's going to be dependent on the prism that you're using, and so for our prism, it's about 200 nanometers, and we're spreading that out amongst about 16 pixels, and so we get it's close to 20 nanometers per pixel.
Did you see the question? Okay, thank you. Next question, are you doing demos at various locations? Yes, we are. We have, in Europe, have a couple of one demo system in Europe. We'll have a second one over there later this spring, a couple of demo systems in the U.S. We've been actively lining up demo locations for 1 to 2 week demos. If somebody's interested in having a demo of this system, they should certainly contact us, and we'd be happy to arrange that. Question about the sensitivity of the detector array. Is it also comparable with conventional detector or the weak signal?
So for traditional detection of spectral signals, people have been using PMTs, which is about 30 to 40% quantum efficiency. But here, the main reason why this is more sensitive than those systems is because we're using a EMCCD camera, which is usually around at least greater than 90% quantum efficiency across the visible spectrum. And that coupled with the prism that we're using, which is greater than 90% transmission, we're getting much more sensitivity in our samples. So.
Yeah, sort of anecdotally, one of the people that we've done a demo with this system, the group at the NIH, the person there that does their software, their image processing, and has done a lot of work when developing unmixing algorithms. That was one of his first comments when he started doing some analysis or processing on the data. He was quite impressed with the signal-to-noise ratio of the images that we're generating on this system compared to what he's been seeing off of a traditional point scanning system. Did you say 200 nanometers? That was with respect to your spectral spread. Yeah, it's 200 to 220 nanometers. It, to some extent, depends upon, we typically put a wide bandpass filter in the light path between the scanner and the camera so that we're filtering that bandwidth, and it typically ranges between 200 and 220 nanometers. We're at the end of the list of questions. If there's any other questions, please send them in. We'll wait a couple minutes, see if anything else comes in. Well, if there's no more questions, that's the end of our presentation today. We thank you for your attendance, and we look forward to chatting with you again sometime in the near future with another webinar. Thanks.